I am Salim Uzgrin from Harmony Public Schools, and my PBL project is on genetics.
Before I begin, I would like to thank Mr. Serdar for helping me choose my topic. I would also like to thank Mr. Reese for editing and revising my script, and Mr. Toledo for helping me with my experiments. The driving question of my PBL project is, how do genetic crosses provide information about inheritance? This Punnett square is extremely important for making predictions about inheritance. In this picture, the uppercase G is dominant to the lowercase g. Both the parents are heterozygous. The offspring, also known as F1 generation, are one homozygous dominant allele, two heterozygous alleles, and one homozygous recessive allele. If this cross between both heterozygous parents are experimented on, the Punnett square predictions will be fairly correct and rarely incorrect, according to many scientists' investigations. The materials needed for my genetics project are 4 coffee filters, 4 petri dishes with covers, 4 plastic mini bowls, a fluorescent light source, a water dropper, 10 hairy purple stem seeds, 10 hairless non-purple stem seeds, 10 F1 generation non-purple hairless seeds, and 10 F2 generation non-purple hairless seeds. After many countless nerve-wracking failures, I ended up starting my project for a fourth time. This was incredibly frustrating since everyone was ahead of me, and I was really behind. The fifth time I had planted my seeds, it looked like I had no problems. I saw a glimmer of hope approximately 6 hours after I planted my seeds. The stem was slowly breaking out of the seed. After 12 hours, the leaves began to grow. 24 hours later, the plants were halfway grown. 12 hours before my plants were fully grown, the stems were already visible. Finally, my plants were fully grown. I did my Punnett square and the predictions I made about my plants were surprisingly all correct. That answers my driving question in which I had to evaluate whether Punnett squares give accurate predictions about inheritance. For my ELA component, I had to document 9 journal entries for every quarter of the school year. I had to do 36 journal entries overall for my PBL project. In each journal entry, there had to be 8 through 9 sentences each, and each had to be grammatically correct. For my social studies component, I had to have a 2-paragraph brief history about genetics. I also made a 20 event timeline about the major events in the history of genetics. Thanks for watching, Harmony Schools.